Hello everyone, and welcome to Towards Sub-Cellular Mass Spectrometry Omics, the SEISMIC facility for spatially resolved single and subcellular omics. I'm Matt Thorne of LabRoots, and I'll be your moderator for today's event. This educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. We do encourage you to participate today by submitting any questions you have during the presentation. To do so, you can simply type them into the Ask a Question box and click Send. We'll try to answer as many questions as we have time at the end of the presentation. And you can also submit any technical issues you have there as well if you're having any trouble seeing or hearing the presentation. I'd now like to welcome our speaker, Melanie Bailey, professor at the University of Surrey. For a complete biography on our speaker, you can click the biography window on the left side of your screen. Melanie, you can now begin your presentation.
Hello, my name is Melanie Bailey. I'm a Professor of Analytical Science at the University of Surrey, and I'm here to talk to you today about the wonderful world of spatially resolved single and subcellular omics. So why single cells and why spatially resolved? Well, human disease starts within cells and spreads spatially. So if we really want to understand how cancer develops, how infectious diseases manifest and how to treat them, we really need to be studying cells one by one. And if we do bulk analysis, where cells are very heterogeneous, we don't see those spatial effects. And so there's been a huge driver in the last decades for spatially resolved and single cell techniques. And in fact, the market is predicted to be worth 7.6 billion by 2033. So at the SEISMIC facility at the University of Surrey, we have various different techniques that can be used to isolate and probe the spatial distribution of analytes in single cells. So we have various different approaches for mass spectrometry imaging, including some very new, exciting and bespoke approaches using an ion beam accelerator. We have laser capture microdissection, which you can use to cut around cells, either in tissue or in culture. We also have microfluidics, which will rapidly isolate cells one by one and put them into 96-well plates or vials. And we have capillary sampling, which is a technique that has recently been commercialized by Yokogawa and is the focus for the SEISMIC facility, which I'm going to describe in more detail. So the SEISMIC facility has been set up as a national facility in the UK. It's a collaboration between different bioscientists and people that can do multi-omics and people that can do capillary sampling. And the idea is to develop single and subcellular omics techniques and disseminate them to the wider bioscientist community within the UK. So there are about 30 different universities and industry and academic institutions who are involved in this collaboration. So the SEISMIC facility is using and adapting instrumentation from the Yokogawa Corporation, which has recently been commercialized. And this is a really cool innovation that allows you to sample just a single cell or a part of a cell under confocal microscopy. So the cells are alive at the point of sampling, and there's a nanomanipulator that drives a capillary down to sample just one cell, retaining the position of the neighbours. And as you can see here, you can actually see the cell being aspirated into the tip, leaving behind the neighbouring cells, which allows you to start to look at spatial effects or explore the relationship between cell morphology and omics markers. We're also able to sample within cells. So here you can see sampling of nuclei. This is the before and the after image of nuclear sampling. We, with Yokogawa, performed sampling of nucleolus within a cell. So here you can see a nucleolus going up into being aspirated into the capillary tip, leaving behind the neighbours. You can see there. And then on the right, you can see sampling from the mitochondrial region of the cell. So we pierce through the cell membrane, we aim at the mitochondria using the confocal microscopy, and we aspirate material into our tip. On this slide, you can see sampling of intracellular bacteria. So we're actually sampling the phagosome within a macrophage into our tip. And then on the left-hand side, you can see that we're sampling within a cell these subcellular stress granules that form when a cell is stressed. So basically what we've got here is a platform technique for being able to extract either a cell or a part of cell into a capillary. And then once you've got that material captured, you've got all sorts of opportunities for downstream analysis. So you could look at elements, metals, lipids, proteins, transcripts, genes, and metabolites. So it's a really interesting analytical challenge, and we've been looking at how to couple capillary sampling with various different mass spectrometry techniques. So we've very much been focusing on the analysis of samples using mass spectrometry. So you've got your material captured into your tip. What do you do with it? Well, the most straightforward way to analyse that material, because you've captured it into a nanospray tip, is to perform nanospray ionisation. So what you would do is backfill your capillary tip with some ionisation solvent and spray the analytes directly into the mass spectrometer. But that carries a few disadvantages because all of your analytes are being ionised all at the same time, so they're all competing with each other for ionisation. So you get problems with quantitation, you can get signal suppression, and of course you can't resolve any isobaric interferences. So we've been exploring transferring the cell into a liquid chromatography vial and performing LCMS. So on this slide, you can see our reasons for using LCMS, and nanospray ionisation can be really good for looking at targeted analytes, but in our experience, we've found that when you don't have an internal standard that's isotopically labelled for each of your analytes, so you're doing an untargeted analysis, nanospray really lacks precision compared to LCMS. The other issue that we've had is that it's very difficult to get the internal standard and the cell material to co-locate in the tip, because what you get is a backloaded internal standard and a cell that's front-loaded, and it's actually very difficult to get the two to mix into your capillary tip. And so what this means, as you can see for the plots on the right, is that it can be quite difficult to separate samples from different groups. So this is a single cell metabolomics analysis with nanospray ionisation on the left and LCMS on the right. And you can see with the LCMS data, it's just much easier to see the difference between different groups. So we actually started this work several years ago on cells that had been spiked with anti-TB drugs. And we were actually able to develop a method that gave us a linear response to drug analytes. So we were able to quantify in picograms per single cell the level of drug uptake. And we actually compared cells with high and low drug uptake, and then we looked at unknowns. And we did a PCA to see what the differences were between high and low drug uptake. And we were able to see that some of the analytes that we were detecting, some of the features could be tentatively identified as lipids, and that prompted us to try to develop a method for single cell lipidomics. So our early work in this area used an analytical flow method. We took some help from Thermo to develop it. We used a C30 column, which gave us very sharp analyte peaks for good sensitivity, and we were able to see that firstly and importantly, cells and blanks were different, and we were able to detect around 400 lipid features per single cell. But these lipid features, we didn't necessarily, all we had was an M over Z value. We didn't necessarily have a database match, and we certainly didn't have very many lipid features with fragmentation spectra. So we were only getting around 5 to 10 the lipid features per single cell with fragmentation spectra. But nonetheless, we were actually able to show, and we published this work in Analytical Chemistry in the paper that you can see presented on the slide, that cells that were dosed with the chemotherapeutic drug gemcitabine had different lipid profiles measured at the single cell level compared to control cells. And actually the differences that we saw in lipids between dosed and undosed cells matched what had been seen in literature in bulk. Now, of course, this isn't why we want to do single cell analysis. We don't want to just repeat what has been done in bulk, but obtaining an initial validation of the method and showing that the methodology can replicate what has been done in bulk is very important. So on the subject of validation, we've recently published a paper in Analytical Chemistry which compares two different methods of sampling single cells and exploring the similarities and differences in the lipid profiles between sampled cells and blanks. And here you can see the lipidomics data that we're getting with our analytical flow data methods in two different methods, completely different methods of single cell sampling. So we have microfluidics, which is a technology from IOTA that we have managed to develop to perform single cell lipidomics compared to our capillary sampling method. And you can see here the differences between blanks and capillary sampled single cells on the left compared to microfluidic sampled single cells. Now you can see that there's first of all significant differences between the lipid profiles of blanks and cells, which is extremely encouraging, but also what we're seeing is that the capillary sampled single cells and the microfluidic sampled cells, we have a lot of heterogeneity, which we expect, but we're also sort of seeing broadly similar lipid profiles, provided we do the blank correction appropriately. So this comes me on to the next question of, are we really measuring cell lipids? And one of the things that we've been extremely concerned about, or sort of wanting to validate, is whether we're sampling the cell or the cell media. What really is the measurement when you do capillary sampling? Because when you sample a cell, you do actually aspirate a small volume of cell media alongside your cell. And so Anastasia, my PhD student, has been looking at different methods of single cell sampling and also whether or not we can see a difference in single cell lipid profiles between cells that are sampled in PBS or cells that are sampled in media. And you can see on the PCA plots on the right that our different cell samples look very similar and they look different to the blank samples, provided we do the blank correction appropriately. What we also see is that if we deliberately pull up too much PBS into our tip alongside the cell, the number of lipid features that we see reduces because we're starting to see ion suppression from that PBS. So moving on, we wanted to see if we could improve the confidence of our peak annotation with our single cell LCMS method because, as I mentioned previously, at analytical flow, we were getting very, very few features that we could generate MS2 spectra from. So the way you can tackle that, of course, is that you can take a bulk sample and you can try to match your retention time and your accurate mass in MS1 to your single cells to your bulk. But we really wanted to try to see if we could collect MS2 spectra from single cells to increase our confidence in peak identification. And so we actually we worked with SciEx to use a Xenotoph mass spec, and we scaled our method down to microflow, and we were actually able to demonstrate for the first time that we could collect fragmentation spectra for single cell lipidomics by LCMS. And again, that work is published in Analytical Chemistry if you want to go and have a look. So we've been working with various different mass spec vendors, so Thermo, Bruker, SciEx, Waters, to see what the best-in-class mass spectrometers can do, how can they perform for single-cell lipidomics, and we've been shipping single-cell samples for analysis. And perhaps unsurprisingly, as you go down to nanoflow rates and as you use more and more powerful mass spectrometers, you can see more and more lipids per single cell. So we get from around 100 lipids per single cell of the ones that are represented in our internal standard to around 400 lipids per single cell when you use the most powerful mass spectrometers on the market. So we've been working with Rahul Deshpande and Susie Bird from Thermo in the US to see what some of the more state-of-the-art approaches and nanoflow can do for single-cell lipidomics. So we've been doing this using the Vanquish Neo and Orbitrap Exploris 240 mass spectrometer. And amazingly, when you scale down to nanoflow, you can get really nice single-cell lipidomics coverage. So we're able to see over 400 lipids per single cell. And what I should say here is that this is quite a restrictive analysis. So we're restricting ourselves to features that are represented in our internal standards, so only lipid classes that are present in our EquiSplash internal standard. We've already got a 50% feature filter, so it has to be present in 50% of our samples to be considered detected. And we've also applied a quite strict blank correction from our media. So what's really nice about this method and this mass spectrometer is that it's fast enough to perform positive negative mode switching. And what that enables us to do is, first of all, expand our lipid coverage, because we see some lipids in positive mode only, some lipids in negative mode only, but also to confirm our peak assignment, because in positive mode we will see certain characteristic peaks in the fragmentation spectrum and other characteristic peaks in the negative mode fragmentation spectrum. And while single cell data is quite sparse, we don't necessarily see many fragment peaks. When we put the two together, we can actually get a great deal of information, which enables us to confirm our peak assignment. So now I want to show you an application of our methodology that we performed in collaboration with Thermo. So this actually combines imaging, single-cell lipidomics, and spatial information, and really shows how the single-cell level information combined with spatial information can start to help us to decouple new information in biology. So we here are looking at pancreatic cancer. And pancreatic cancer has a really low survival rate, and it causes problems because of radiation sensitivity. So cells that survive radiation become much more resistant to treatment, but the mechanisms of that radiation resistance are unknown. And so the question was, can single-cell lipidomics help us to understand these mechanisms? So one thing to point out here is that we are starting to explore the radiation-induced bystander effect, and this is a considerable parameter, I guess, in treatment optimization, because if you hit a population of cells with radiation, they become stressed, but the radiation injury can also induce stress in neighbouring cells, and of course that has implication for patient treatment. So what we did in this experiment to explore radiation-induced bystander effects was that we irradiated cells through a pinhole in lead shielding. So we had a sort of a letterbox hole over a cell dish, and we irradiated cells through the hole, leaving the neighbours untouched by radiation. And then we went in with our capillaries and we sampled cells that were directly irradiated, their neighbours, and cells from a control dish. And we actually prepared those cells and shipped them, individual single cells, to Thermo for analysis, and we actually reserved a batch of cells also at Surrey for a validation experiment. So first of all, we were able to see differences between the lipid profiles of irradiated cells that were directly hit by radiation and the control cells. So we're seeing overall a reduction in lipids, but also specifically in lipids that contain polyunsaturated fatty acids, particularly those that contain arachidonic acid. And this is interesting because cells that have low polyunsaturated fatty acids are more likely to survive oxidative stress that is brought on by radiation. Now, what was really interesting in this work was that we found that bystander cells and the cells that had been directly hit by the radiation were indistinguishable, but both were different to the control cells. So it's like both cells are responding to the radiation, even the ones that were not directly hit. And we actually performed analysis of the lipid droplets that formed in on the same set of cells that we extracted for mass spectrometry analysis. That's one of the nice things about this technique, is that you can explore the cell morphology on the same cells that you extract for mass spectrometry analysis. And we were actually able to see that the measurements of lipid droplets corroborated our mass spectrometry data, because the lipid droplets increased in size and in number after irradiation, both in the directly hit cells, but also in the bystander cells. So this is really interesting. We've got mass spectrometry data and lipid droplet data that corroborate each other based on our samples that we've sent to Thermo. But what's even more interesting is that the validation set of data, so the cells that we ran at Surrey as a validation experiment, actually found very similar trends to the samples that we sent to Thermo. So we think this is really exciting. This is the first time that anybody has explored differences in lipids after cell irradiation spatially. And it's also the first time anybody has attempted a single cell validation experiment where two different labs have analysed the same set of cells and been able to detect the same biological response. So, as I mentioned previously, there are various different ways that you can analyse the material after capillary sampling. And as well as looking at lipids, we've also been exploring the coupling of capillary sampling to other analysis methodologies. And next I'm going to talk to you about the coupling of capillary sampling to elemental analysis through laser ablation ICP-MS. So this is some work that we carried out in collaboration with King's College London, and our collaborators there were very interested in trying to establish the delivery of radionuclides to particular compartments within the cell. So they're trying to deliver thallium into the nucleus or mitochondria of cells to kill cancer, but currently they're unable to confirm the subcellular localization of that radionuclide. And so the aim of this experiment was to see if we could detect thallium at in subcellular compartments. And so we did this by trying to couple capillary sampling to laser ablation ICP-MS. So here you can see some of our experimental setup. So we use a manual apparatus, home-built apparatus, to transfer our capillary sampling material onto a silicon wafer. The aim is to try and make as small a drop as possible and then ablate the entire drop of material that we elute from our capillary and transfer those analytes into the ICP-MS. And so we worked with various different substrates and internal standards and elution protocols to optimize this methodology. And eventually we were able to demonstrate that we could detect thallium in whole cells. Now, that wasn't really the purpose of the analysis, but it shows that we have enough sensitivity. The next step has been to go subcellular. And so here you can see the removal of mitochondrial material, so it stains the mitochondria in the cell, and you can see the before and after picture. You can see that we have been able to remove material. We can confirm that we have removed that material by looking at the tip. Under the microscope, we can use the confocal to show that the sampled material is in the tip. We can then elute the material and look at the tip again after eluting. Now, we don't have 100% recovery, but what we are actually able to do is by depositing that sample on a surface, we can actually detect thallium in our subcellular samples. So the technique needs perhaps some optimization. There are a few questions about how spatially selective capillary sampling is, but this is the first time that anybody has been able to go within a cell with a capillary and couple it to ICP-MS, and we think that that is very exciting and offers lots of interesting possibilities. So the last thing I want to talk to you about is single-cell proteomics. So I'm sure you're aware that single-cell proteomics has come on leaps and bounds in recent years. The coverage of the proteome that you can see at single cell level has increased dramatically, and most single cell proteomics methods don't provide any spatial information. And of course, with capillary sampling, as I've shown earlier, you can obtain spatial information and morphology of the cell, and of course you can also go subcellular. And so coupling capillary sampling to proteomics could offer all sorts of exciting applications in biology. So we sought to couple capillary sampling with proteomics, and we started this work with Ed Emmett, who's from the University of Liverpool, and by sampling cells using our methodology, shipping them to him for sample prep and analysis using a label-free workflow, we were actually able to see differences between two different cell lines and blanks using this methodology. Now, certainly the coverage is a little lower than we might have anticipated, probably because of the shipping and also because of potential loss of analyte in the capillaries and a lack of optimization. So next we worked with Nicholas Locker, who is from the Bragg Institute, which is just down the road from us in Surrey, and he's exploring stress granules that form within cells upon virus infection, and what he'd really like to be able to do is characterize the proteins that exist within the stress granules. And so what we were doing in this work was to explore the differences between whole cells that contain stress granules, capillary sampled stress granules, and unstressed cells. And so what we did was to pick capillary sampled stress granules and whole cells from the two different groups. We deposited them into plates and we shipped them cold to the group of Erwin Schoof, who are based at Technical University Denmark, and they have some amazing methodology and instrumentation for single-cell proteomics using some of Thermo's newest workflows and instrumentation using the Orbitrap Astral. And what you can see here on the left is that, first of all, the integration of capillary sampling with single-cell proteomics methodologies has been successful. We're actually able to see differences between stress granules, between stressed and unstressed cells. They actually cluster according to their protein content. There's certainly a lot of work that we need to do to optimize our technique, but it's very exciting and a first demonstration that we can actually do subcellular proteomics. So just a few words in conclusion. We're developing single and subcellular capillary sampling and coupling this to LC-MS, but also to laser ablation ICP-MS. LC-MS, in comparison to nanospray, can reduce measurement variability for for single cell analysis, and it can also aid peak assignment, which is why we're using it. With appropriate blank correction, we think that capillary sampling and microfluidics can give similar lipid profiles. We can perform lipid droplet analysis or cell morphology analysis using live cell imaging, and then follow that with single cell lipidomics, which we've shown in this talk. And I think the other thing that is exciting about the work that we've done recently is that we've been able to show that we can actually ship samples to other laboratories for analysis, and we can still see biological responses that we can replicate back in our own lab. And so if you're listening to this and you have a new methodology that you'd like to try, perhaps you have a way of analysing capillary sampled material, please do get in touch. We'd love to hear from you and work together to see what we can do. And then finally, we can couple capillary sampling to elemental analysis. So I would just like to thank all the sponsors of this work. I'd like to make a final note to advertise the work of Analyst, the journal. This is the Royal Society of Chemistry's longest running journal, and we're celebrating our 150th birthday this year. So I've just taken over as the editor-in-chief. We've got a number of different special issues coming out this year, and if you're working in the area of analytical science, which I assume you are if you're listening to this talk, I hope you'll consider publishing your work with us. And finally, I'd like to thank everybody in my group and the sponsors of this work and our collaborators, particularly Thermo, who have been incredible at giving us help and assistance to develop our methodologies. And thank you so much for listening.
All right. Thank you very much for that very informative presentation. We'll now go into the live Q&A. So if you have any questions that you haven't sent in yet, please do so now, and we'll try to get to as many as we can today. So let's start with, you've emphasized the importance of validation for single-celled lipidomics. What are the next steps to further improve interlab reproducibility and confidence in single-cell measurements, especially for subcellular compartments?
Yeah, thank you. It's a really good question. It's a really difficult topic because, of course, the reason that we're trying to do single-cell lipidomics is that cells are heterogeneous, right? So you have cells that are heterogeneous, you've got no standards. So how do you actually validate the methods? So, you know, I guess we've taken steps in that direction. So we have performed our microfluidics versus our capillary sampling comparison, and we've been able to show that we can see different lipid profiles with two different techniques. We've performed our interlab comparison with Thermo to show that two different labs can see the same biological response with two different methods. And I guess steps in this sort of further direction would be to perhaps develop more targeted and quantitative methods to see if laboratories can reproduce their analysis at the single cell level. And at the subcellular level, there is so much more work to be done. So particularly around how spatially selective those methods are. So when we're picking out a subcellular compartment, are we really targeting, for example, just the mitochondria, or how much cytoplasm are we picking up? How much cell membrane are we picking up? So all of these things are challenges for the future.
Thank you. Let's see here. You mentioned ion suppression when excess PBS is aspirated with the cell. Are there any strategies being developed to minimize this or correct it for during analysis?
Yeah, so I think what we were trying to do there was to show whether, because we know that we aspirate some submedia or contents that the cell is sitting in alongside the cell. So we were trying to determine if you try to do your blank correction properly, does it matter whether the cell is sitting in PBS or whether the cell is sitting in media? And I think what we were able to show was actually we don't see any difference in lipid profiles, provided you do the blank correction appropriately. When we pulled up too much PBS, we did that deliberately to try and make the measurement go wrong, and then it does go wrong. But I think what was nice about it was that we didn't see more lipids, which you would expect if the lipids are actually coming from the PBS and not from the cell. We saw fewer lipids. So it's a sort of a validation that what we're seeing is coming from the cell and not from the surroundings. So we are looking at how much material we're capillary sampling, because of course you can adjust the different pressures and the Z position of the capillary when you apply the pressure, to sort of control the amount of media that is being aspirated with the cell when you sample it. So we're doing some experiments with fluorescein so that we can actually see the amount of media in the tip versus cell, to try to reduce that contribution from the media and therefore our blank correction.
All right. Next one we have is, given the challenge of limited database matches and the MS coverage in single-cell lipidomics, what do you think is needed at the community level? More comprehensive libraries, better spectral acquisition methods, or something else?
Yeah, so I guess I am a fan of being able to collect the MS spectra from the cells themselves, so that we don't have to resort to, making big libraries to annotate our cell lipids. So some of the newer approaches that we've been working on, particularly with nanoflow and microflow, enable us to collect those MS2 spectra from the single cells themselves. And I think that's got to be the way forward, particularly because cell populations are so heterogeneous. We really need to be trying to collect that MS2 spectra from the cells. So I guess what we're looking at is faster and more sensitive acquisition methods in answer to the question.
Next we have, you noted that spatial selectivity of capillary sampling is still a question. What are the current methods or controls used to assess or ensure sampling accuracy within specific cell regions?
Yes, that's a really good question. So what we are currently doing is we have a bright field image and a video that's taken when we do our subcellular sampling. We can stain all different components of the cell using fluorescent dyes, and then we can use confocal microscopy before and after we pick the cell to get intelligence on what has been sampled. And we can also do confocal microscopy on the contents of the tip, so we can get an idea of what has actually gone into the tip. We can elute the material from the tip and then look and see what's left as well. So by using that information, we can optimize the components that we can visualize. Another way to look at it is to look at housekeeping proteins. And so now that we have established methodology for sub-cellular proteomics, it's possible that we would be able to look at housekeeping proteins from different subcellular compartments to enable us to sort of guide our optimization.
All right, excellent. Let's see here. Do you see a path for using capillary sampling and LC-MS workflows in clinical diagnostics or personalized medicine, especially for drug response profiling at the single cell level?
Yeah, absolutely. So I think one of the things that capillary sampling gives you that other single cell sampling techniques don't give you is the spatial dimension. So I guess it gives you the context of morphology, or a cell that has been subject to a particular biological stimulus, perhaps something spatial like radiation, like I showed in the example, or perhaps a cell that is sitting next to an infected cell. I think it will give you that kind of information, those cell-to-cell communication mechanisms that you're not going to see with microfluidics sampling techniques. They're much higher throughput, actually. So, if what you want to do, if the purpose of your analysis is just to explore the relationship between drug uptake and omics markers, then actually you might be better off using a microfluidics method. But if the purpose of your analysis is to explore spatial effects, then I think that's where capillary sampling really comes into its own, or morphology. So if you want to relate cell morphology to particular lipids or metabolite signatures, or you want to go subcellular. And then in terms of clinical applications, it's interesting you should say that. So my group are going to be moving to King's College London in May, and we'll be in Guy's Hospital. So I'm really looking forward to being able to explore more clinical and translational applications of the technology. And I think there's all sorts of exciting things that we can do in cancer and infectious diseases, across the board.
All right. Let's see. Shipping single cells for analysis is impressive. What are the critical factors that enable success there, and what advice would you give to others hoping to collaborate remotely on similar studies?
Yeah. So I guess that's one thing I tried to bring across in my talk, actually, is that there aren't many labs that have this kind of technology, and we're very open to the idea of collaborating with other labs. We've shown that we can send samples overseas now. I guess one of the things I think is attractive about capillary sampling is the fact that there's very low dilution of the cell material, the cell contents. We found that for lipidomics, freeze drying and then shipping the sample at room temperature and then reconstituting the sample in the third-party lab does partially change the single-cell lipid profile. But as we showed in the comparison that we've done with thermo on radiation effects in single cells, we were actually able to still replicate the same biological response to radiation. So that's certainly suggestive that we can send freeze-dried cells between labs, and that's certainly not been done before. And then for proteomics, what we've been doing is depositing cells into plates and then carrying out cold transport of the cell materials. And as soon as they reach the lab in Denmark, they are digested and sent off for analysis.
All right. Looks like we've got time for one more question. How do you inject the sample for single-cell lipidomics? In the setup shown with Vanquish, NEO, and Exploris, do you use microbore, cap-LC, or nano-LC column? What kind of source is needed in this case?
So Thermo used an easy spray column and an easy spray nano source, which was connected to the Vanquish Neo in this case.
All right, excellent. I'd like to once again thank our speaker for your time and important research. We'd like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's webcast. Before we go, I'd also like to thank the audience for joining us and your questions. Any questions that we didn't have time for today and those that are submitted during the on-demand period will be addressed by the speaker via the contact information you provided when you registered. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available. We do encourage you to share that email with your colleagues who might have missed today's event, So until next time, thank you very much and goodbye.